Thank you for joining us for our Advancing Research with Linear Probes webinar series. Today's webinar is the last webinar in the 5-part series. And if you're interested, the previous webinars are available on demand at plexon.com. And following today's presentation, we will hold a live question and answer session. So please use the Q&A feature in Zoom to enter any questions that you might have for the speaker, and she will address those at the end of the webinar. Today's speaker is Ariana Andrei. She is a postdoctoral research fellow at the University of Texas Health Science Center in Houston in the Gergoy Lab. She studied film physiology and neuroscience at York University, McGill University, and the University of Texas. Her work uses a combination of electrophysiology, optogenetics, and a lot of computers to study how the brain generates sensory perceptions. Are you able to take the screen share?
Yeah, working on it. There we go. All right, everyone can see that? Okay. So thank you, Naoko, for the introduction, and thank you everyone for coming after a small 2-week delay. So I'm going to tell you about optogenetics using laminar electrodes. So to begin, I'm going to give you a really brief background on just visual processing and the architecture of V1 that is important to understand the scientific part of the talk. And then I'm going to tell you about generally using laminar electrodes and combining it with fiber optics. And we're going to look at how the perceptual impact of activating excitatory neurons using channelrhodopsin. And then if there's time, I'll tell you some experiments where we're actually silencing. So the second part is we might not get to it. And I've also included a lot of technical details because I find that over the years, most of the questions I get are really technical. So I figured this would be a great forum to just kind of share that insight. Okay. So visual cortical processing is really a distributed and hierarchical process. So by that, I mean, you have all sorts of visual areas spread out in different parts of the brain. And you have each area kind of representing visual stimuli of increasing complexity. So you have receptive fields in V1. So this is a representation of receptive field size in V1. And the center part is near the fovea, and this is the periphery. So what you can see from this diagram is that receptive fields kind of tend to increase towards the periphery. And what you find is that you have small receptive fields in the fovea. And these cells in V1 are interested in pretty much like very rudimentary sensory stimuli, things like oriented ratings, luminance, contrast, that kind of thing. And as you move up the visual cortical hierarchy, you have receptive fields that increase in size. So from V4, so V2 to V4, you can see that the receptive fields get bigger. And also the properties that the cells encode become more and more complicated. So you have cells that suddenly represent things like contours, colors, and eventually you get to places like IT where you can get whole object representations. So this is kind of the history of neuroscience for the visual neuroscience for the last 100 years or so. We've kind of mapped out all these different stimulus features that are represented across the brain. Perceptually, though, the problem kind of sums up to this little drawing of mine here, not mine, this little prism diagram. So what we know is that the brain represents all these different visual stimulus features. We have orientation, luminance, contrast, color, motion direction, depth, all spread out across a whole bunch of different brain areas. However, if you're watching this video, you kind of realize that visual perception itself is actually a very cohesive experience. Somehow all these different components are mashed together into like what we see. Now, the question is, how does that actually happen? What are the rules by which all these different stimulus features are combined? Going across stimulus features is a very complicated problem. So what we're going to do is break it down a little bit. So we're going to focus just on orientation in primary visual cortex. So something that's really well studied. Okay, so this is just a little bit of functional architecture of V1 in primates and carnivores. So this is different than rodents. That's why I bring it up. So probably neuroscience 101, if you take an electrode and you penetrate the cortical surface and you go perpendicular to the surface, you find that cells have overlapping receptor fields, and they also have very similar orientation to it. If you, on the other hand, if you could manage this penetration, what you'd find is that you have a representation of all orientations across adjacent columns of cells. So you have, what's interesting is that this orientation information is actually systematically aligned across the surface. So you have cells that are orthogonally tuned or actually further apart than cells that are more similarly tuned. Okay, so this is pretty much what you need to know to make sense of some of what I'm going to tell you later. Okay, so now going into V1, if you can imagine you send a visual stimulus to V1, and you have a bunch of cells or a whole population of cells that are tuned to the same patch of the visual space. Any stimulus that you present is actually going to activate a whole bunch of cells in really different ways. So the cells that are tuned to that particular orientation, let's say this part of the lionfish, you're going to get a strong activity. Ones that are tuned, say, if you're on the flank of these tuning curves, you're going to get a weaker response. Whereas if you're in the non-preferred orientation, you're going to get just baseline activity. So somehow the visual system is able to combine all these signals and to produce your sensory percept of this little patch of visual space to begin with, but also eventually of the whole image. So the question is, how does this pooling happen, and what are some of the what is a rule, at least, by which this occurs? Okay, so in order to address this question, we're going to have our animals do a detection task. So the animals will fixate. We're going to present an oriented grading of some different orientations, different contrasts. And then the animals are going to respond, whether or not they saw something. And then what we're going to do is we're going to inject an artificial signal, which is the optogenetic stimulation on half the trials. And we're going to see how that artificially injected signal actually interacts with the natural stimulus-driven activity. Okay, so this is where that architecture of V1 comes into play. So essentially what we were trying to figure out was between two different possible hypotheses. So because it's a detection task, there's two ways you could solve this problem to pretty much signal that there's something on the screen or not. You could, so the gray, this little mountain of activity is a stimulus-driven activity due to the visual stimulus on the screen. And this blue mountain of activity is what we're going to inject optogenetically. So if you're doing a detection task, you could imagine just summing both of these signals. So near the preferred, either in this case or this case. So this little mountain plus that little mountain equals the same overall change in activity across a small population. So if behaviorally you find that regardless of the orientation tuning of the two populations, that your signal is effective, if your perceptual activity, if your change in behavior essentially is modulated regardless of what the orientation preference of the two cells, you could say that all the local signals are being pooled. However, if you find that the perceptual activity is only modulated in one condition or another, you could rule out, you could say that mostly the local signals or the distant signals are somehow pooled. So I'm going to talk about near and far, but near and far are essentially like orientation space, which in macaques and carnivores and other primates, it pretty much translates to a physical space across the cortical surface. Okay, so these are our two options. So either local signals are pooled, which is this near condition, or all signals, so regardless of the orientation preference. Okay, so now we're going to get to the technical part. So we're going to talk about how do we actually inject that signal. Okay, so now we're gonna use channelrhodopsin, and this comes in a viral vector preparation. So all you do is inject it into the brain. So we're using a standard chamber setup, and then we had this custom grid made. That'll come into play later. So the grid gets mounted on top of the chamber. And then what we did is we rigged our microdrive system to advance, instead of an electrode, we're gonna advance a syringe. And then we actually rigged our microdrive so I should be able to push the plunger on the syringe as well. And then what we did is we knew the depth at which we found cells. So what we did is we went to the lowest depth first, and then we injected 1 microliter in a column, very slowly. And then we did this for multiple columns in the general vicinity. The idea was to create a large enough target area so that we wouldn't have to do too much fishing when it came to actually trying to find the transfected cells. And then once you do this, you just wait a few weeks for the virus to express. The important thing is to make sure that you don't change your grid to chamber arrangement so that when you come back in a few weeks, you can actually locate that site. Okay, so now we're actually going to activate the neurons. So this is what one of our fiber optics looks like. Because we're trying to modulate behavior, because we want our final metric really is a behavioral measure, we really need a lot of light. So this is why we opted to go for the fiber optic instead of having the embedded ones. Also, I think at the time when we started these experiments, the embedded fiber optics inside of the U probes weren't available yet. Okay, so now what we're going to do, so now you can see the grid has these little, it looks like a little figure 8. So the idea was to have very close spacing between the fiber optic and the U probe is what we used here. Now, if all goes well, this is typically actually the arrangement where we had the fiber optic aligned to the top of the U probe and kind of shining light essentially in front of the contacts. So what we find is that if all goes well, you'll shine your laser. So you'll use like a pulse laser stimulation, you get really nice laser responses, like unambiguous, very clear. And then if you look at the change in firing rate, as I'm showing here, between just fixation versus the laser condition, and this is time, and these are the contacts across the probe. So you can see at least across these middle contacts, you get very strong activation. It's so strong and clear because this is the original channelrhodopsin, so it's got really millisecond timescale precision. You can pretty much count the laser pulses very clearly, but you can also see these little trails of activity that I think are due to more network-based effect, as that activity you're sending in actually propagates throughout the network. Okay, so sometimes this might not work as well as you'd hope. The major problem is that light, particularly blue light, scatters a lot inside the brain. So this is from a paper from some years ago now. This is blue light. This is in saline. So you can see light in saline is like a blowtorch. It travels very, very far. However, inside the brain, you don't really get much spread beyond the diameter of the fiber optic. So this was a 200 micron fiber, and you can see that there isn't really much spread beyond 200 microns. I think this blue line is 1% of the power at the light source. So what we found, sometimes it was better for us to do this, to angle the fiber optic even closer to the U probe. So we had a little bit of wiggle room because you're obviously not to scale. But if you shine it a little bit closer, that helped us get more robust activation. But there is such a thing as too much of a good thing. The one thing you have to be careful of if you're going to attempt this is that you don't want to shine the light directly on the contacts. So there's a few papers on this now. It's something called the Becquerel effect, and it has to do, I don't know the physics of it very well, but there's a lot of papers on it. You can just do it. You can just try this experiment yourself to shine light on the electrode contacts, and what you'll find is that this is just a raw signal. You can see that here they're injecting a 4 Hertz sinusoid, and you can see it modulating, and this is an untransfected animal, so there's no reason why these neurons or the neural signal should respond to light, but it looks like it's responding to light. And if you look at the power spectrum here, you can see that there's a clear peak at 4 Hertz, which is what the stimulation frequency, and then at the harmonics as well. And if you look at the spectrogram, you can see this really like strong power increase right at 4 Hertz. So just something to keep an eye on. So you don't want to, what we typically do in order to avoid this is that we'll have the light shine kind of like across the surface, not at the surface. If you point them right at each other, it's an issue, but if you point them kind of like perpendicular to one another, hopefully the light gets on the cells and then the electrode is recording the cells at the same time. And that seems to work quite well. The other thing is you can hear this artifact. It's more of an issue. You don't usually see it in the spike channels, but you'll hear it in the LFPs. Okay, so one thing that we did that was kind of important is to try and get a handle on how much light rush getting in there, trying to estimate how many cells we're actually activating here in these manipulations. What we did is we looked at at cells that were directly driven by the light. So you can see, I didn't put the laser pulses, but so these couple cells here. So again, this is just the contact number, and then the firing rate change from laser to control trials. So you can see these two cells respond pretty much when the laser comes on. And then what you have is these network effects. So we focus on the cells that have the direct activation. So we define that as within 2 or 3 milliseconds of each laser pulse, you get a robust increase in firing rate. And what we did is across sessions, we aligned the response of the most responsive cell. We called that 0. And then we looked at the spacing between other directly responsive cells and their distance relative to that one very robust one. And what we found, essentially using the U probe as a ruler, because the intercontact distance was 100 microns, so we could pretty much just use it to measure out how far the light spreads in our sampling distance here. What we found was that pretty much within 200 microns, you lose about 50% of the direct responses. And in this case, negative means that it's above, and positive means it's below. So it kind of makes sense that you have this asymmetry, because probably there's a bit more light in the downward direction just because of the way the fiber optic is aligned. So you're probably getting a bit more activation here, but this is the same diameter as a fiber optic. So 200 microns is about where you can expect to get a lot of light. Okay, so back to the experiment. Oh, one thing I just realized, going back for a moment. One of the most common questions I get is, how do we actually penetrate the dura? Because we go through the dura with the U probe. We don't do a guide tube. But if anyone's interested, I can demonstrate afterwards how we actually do that, and to keep pretty much to protect the U probe while doing this. Okay, so back to the experiment. So now what we're going to do is we're going to optogenetically activate one population of cells. So this is the population that we're recording from. And then we're going to activate another population using the visual stimulus. So in the near condition where the light-driven activity and the stimulus-driven activity overlap, so essentially we're activating both cell types roughly, or both. We're activating the same cell population using 2 different methods. So here I show you some PSTHs from either the no stimulus condition and then with increasing stimulus contrast. So you can see that when there's nothing on the screen, the laser, so the laser's in blue and the control's in black, the laser activates the neurons, versus when you have something like a high contrast condition where you have an increase in firing rate due to the stimulus, and on top of that, you also have the laser-driven activity on top. So this is in the near condition when the orientation preferences of the optogenetically stimulated population and the stimulus-driven population overlap. Now in the far condition where the preferred orientation of the 2 populations are different, the cells you're recording from, the optogenetically driven ones, are not going to be driven by the stimulus. So it doesn't really matter on the stimulus condition. But we do have this very strong increase in firing rates, but not the stimulus-driven one. Okay, so one important thing for us to confirm was that the amount of actual light-driven activity in both populations was the same. So what we found was, so the blue is the near and the red is the far, and we found no statistical difference. So we always had a spectrum, but we always had very strong activation. So both low and high, near and far, all these conditions, if we looked at the laser responsive population, was always kind of equivalent amount of drive that we're injecting into the system. Okay, so what happens perceptually now? So these are psychometric curves. So this is the animals, this is 2 animals, their detection performance, and this is the contrast of the stimulus, and this is the percent of the time that they report that there's a stimulus on the screen. So if you're not used to looking at these, 0 is when there's nothing on the screen. This is essentially their false alarm rate. And 1 is our highest contrast stimulus, when they almost virtually all of the time say there's something there. And what we find is that for the low contrast stimuli that are fairly difficult to detect, when we add that laser activity, we find this improvement in detection performance. So it's fairly strong. It's about like 10% improvement. So now the question is, what happens in the far condition? So in the far condition, nothing happens. So this is kind of, goes back to our original hypothesis testing here. What's surprising is that in both these cases, we inject a lot of signal, and especially in the low contrast conditions. If we go back a second, in the low contrast, which are these two, you can see that even in the near condition, there's actually very little stimulus-driven activity. It's a very weak stimulus compared, especially to the laser. However, in the far condition, despite the fact that we have this very strong laser-induced signal, it has no impact perceptually, which is kind of interesting. And then if we stop grouping things into these very coarse near and far, and actually group things more finely across the difference in orientation preference between the population of cells that we optogenetically activated and then the stimulus orientation, what we find is that there's this essentially law of diminishing returns. So the further you get away from the stimulus-driven population, the lower the impact that your laser activity is going to have perceptually. Okay, so what does this mean? Essentially, it means that orientation information in V1 is pooled very locally according to similar orientation preferences. So we would have expected perceptually, so similar to the previous graph, if pooling had been kind of uniform across all orientations, we would have seen kind of uniform increases in detection performance. But that's not what we see. What we see is this exponential decay, which means that orientation information is kind of pooled all together. And somehow, which is kind of interesting, all that extra activity that we've optogenetically injected essentially gets ignored. It doesn't essentially like percolate up to influence the percept. Okay, so I've got a bit of time. Okay, so now I'm going to tell you about another experiment. So the previous one was published. If you're interested, I didn't go into much of the scientific detail. I kind of went to the punchline. But I'm going to tell you another one that's in review now. This is another, so this is kind of a slightly different experiment, but quite similar. So here what we're doing is we're going to actually silence a network. And what we know is that what I've shown is that light is very local. Light doesn't travel very far. But we know that neurons are not really local at all. They actually have lots of lateral projections. And what we wanted to see was whether or not, if we locally suppressed a population of neurons, what would be the effects in the local network? So very local, I'm talking about within a few hundred microns. So most of the time people kind of assume that if you suppress, suppression just kind of dies down. And you're really just suppressing that local. But it's possible that due to these complex network interactions, you might actually get something more heterogeneous in the local network that would complicate any kind of behavioral measure. Okay, so in this case, so we used a similar injection strategy, except this time we injected, instead of channelrhodopsin, we used GTACR2, which is an anion conductive channelrhodopsin. Again, we targeted just excitatory cells. And this time, we're going to do, instead of angling the light source towards the probe, we're going to try and keep them separate. Okay. So a little bit more on the architecture. So at very small distances in V1, within about 300 microns or so, the projection patterns are not orientation specific. So if you look at, so this is superficial layers. If you look at within 500 microns, this should be orientation difference at the bottom here. There isn't really a difference between the soma preferred orientation versus the terminations of its axons, which is not the case if you go further. Then you get orientation matching. So essentially the projections of long distance projections tend to be orientation matched or more similar in orientation preference. Here's another, this is an injection of an inhibitory neuron. And you can see that this neuron kind of pretty much targets all sorts of orientations, not just where its soma is preferred. Okay, so what we do here is first, we're going to just show that GTACR2 works at suppressing neurons. So if we put the light source super close to the fiber optic, again, this is a change in firing rate from the control versus laser. And what you can see with this dark blue is that when the light comes on, you get this strong, long-lasting suppression of activity. And this just has to do with the kinetics of GTAs here too. But the point is that you get long-lasting suppression. Now, what's interesting though, is if we record instead about the light source with the directly activated neurons, if we focus on the probe further away, 3 microns, so we find something quite different. So these are 4 example neurons. Each column is a PSH at a different stimulus contrast. So we have no stimulus of the blank condition and then the high. Now, if we were getting direct suppression, we would expect to see a difference in the blank condition in particular. We'd see that long lasting suppression and firing rates, which we don't see at this distance. So it makes sense because light is very local. So all we're going to be doing is looking at the network effects. However, if you start looking at across when we start presenting a stimulus, we find that in this particular case, you can see that you get some suppression with a low contrast, and it kind of continues throughout. It kind of changes the shape of the PSH. So this particular cell has all suppressive responses, as opposed to this blue one or this purple one that seems to show this progressive facilitation of responses, which is the opposite. And then we have these 2 mixed types in between, which are quite interesting. So we have some cells that are activated at the low contrast and then eventually become suppressed at the high. And then the opposite, where they become suppressed at the low, but then facilitated at the high. And what's interesting is that you can find all 4 of these responses distributed along this probe at the same time. So you're suppressing part of the network. And then what you're showing is how this network is actually modulating the stimulus responses of its neighbors. Okay, so what we did is we fit these 4 responses with a contrast normalization function, which fitted quite well. And then we actually looked at the laminar distribution. So this is another common use of laminar electrodes, where you can do a current source density analysis and identify the boundary between the granular and infragranular layer. And then you can assign all of these responses to their different layers depending on their contact position. And what we found was that in superficial layers, where many of these horizontal connections come, we find mostly the suppressive one. So the idea between the type 1 is that you're suppressing its local neighbors, and then some of that local drive is actually usually used to as part of the stimulus response, because we're suppressing that, we actually just see suppression throughout. Where this kind of gets more interesting is when you go to lower layers, you start seeing more of these exotic responses, where you see that the local network actually very highly heterogeneously modulates neural responses. So we see as we go deeper and deeper, probably with more inhibitory neurons kind of involved along the way, we see these exotic responses become most prominent, where you actually get facilitation during suppression of the local network. Now, we also looked at this behaviorally. And what we found was that, so if we divide, so essentially most of the responses were type 1, then came type 2, type 3, type 4. That's how the naming came in. And what we found was that if we divide our sessions across animals, we look at the distribution of different kinds of responses. If we kind of arbitrarily draw a line at mostly type 1, so 50% or more of the responses were purely suppressive, versus the ones that had more uniform distributions of the other types. What we found behaviorally was that if the suppressive site, if the distal site where we were recording from was also mostly suppressed, again at low contrast, we actually see an impairment in detection performance. Versus in the mixed sessions, where you had a network that's more heterogeneously modulated, we actually saw no difference. So what does this mean? We looked at different ways trying to measure whether or not this was just a sampling bias, because sometimes you might just be recording more cells in one layer versus another. That doesn't seem to be the case, mostly because we did have some sessions that definitely had more of the type 1. So what this means is that the local network, the connections between parts of the local network are somewhat heterogeneous. You don't get necessarily uniform. So if you have stronger, I guess, connections of the upper layers, you might actually get more suppression. Versus if this is a little bit more heterogeneous, you'll get this mixed neural response, but also kind of more ambiguous behavioral responses. So these are things to keep in mind if you're really interested in modulating perception. It's like what you're trying to get is something that's as uniform as possible. Otherwise, that signal, even if you're modulating firing rates quite a bit, the network might be doing things that you're not necessarily aware of. Okay, so kind of wrapping it up at this point. So what I showed you in the first part of the talk was that for perception, information in V1 is pooled in an orientation-specific manner. So similar orientations are pooled, whereas distal orientations seem to be treated as independent. I showed you with this brief tour of suppressive optogenetics that the local network diversely modulates stimulus responses for the adjacent cortical columns. So if anyone's a divisive normalization aficionado, this essentially means that divisive normalization is applied individually to each neuron. So the local network component of that is essentially tweaked for each neuron. So you can't just assume that the local network is going to have the same impact on each individual cell. And then behaviorally, our results demonstrate that probably there's some heterogeneity in the way local networks are connected, and that might also impact your perceptual effects. Okay, so now I just want to thank everyone, particularly Sarun Pajoga, who worked with me with the channelrhodopsin experiments, and Samantha Dibes, who worked with the inhibitory channel GTACR2 experiments.